Hi, everyone. Thanks for joining us today. Just so that everybody knows, there's a Q&A box at the bottom of your screen. If you have any questions, please pop those questions in there. Also, if you run into any technical difficulties, Shane's going to be available to help. So put those technical questions or issues in there as well. And because this is a very common question, the session is going to be recorded. A link to the recording is going to be sent out to everybody's registered email address once it's available, and we're expecting that to be sometime next week. So I thank everyone for joining us, and we're going to have a lot of material to cover, so we're going to go ahead and get started. Today's presentation is really about how we start characterizing and validating different new approach methodologies, specifically artificial intelligence and then in vitro testing within the space of toxicity testing. So Shane, if you can move to the next slide. We've got two great speakers today. The first is Dr. Nicole Kleinsteuer. She's the director for NICEDM, and she brings with her a lot of experience using computational approaches, methodologies, and also the evaluation of in vitro assays to support new approach methodologies. And then we also have Dr. Sean McCullough from RTI. So Sean's a respiratory scientist at RTI, and he uses an air-liquid interface, which he's going to be talking about in his presentation, which comes next. After both of these presentations, we're going to have a discussion with both speakers. So please feel free to ask questions about the larger topic and anything specifically about either speaker's presentations, and we'll get to those. So Nicole, if you're ready, we'll go ahead and hand it over to you because I know we have a lot of material to cover.
Great. Thank you so much, Shannon. And I will, as soon as Zoom allows me to, I will share my screen and pop it into presentation mode. Okay. So thanks so much, everyone, for taking time out of your busy schedules today to join us. As Shannon already said, I am Nicole Kleinsteuer. I'm the director of NICEDM and the executive director of ICFAM. And I'm really delighted to be talking to you today about some of the efforts that we have ongoing across the U.S. federal government and with our broad stakeholder network to validate new approach methodologies from in vitro to in silico and beyond. I'm first going to start with a really quick overview of what I like to call the NAMS confidence continuum. So this shows us that this whole concept of biomedical research and toxicology being sort of separate fields, I think is a real false distinction. And what we really need to advance the field of human relevant science, both for public health protection and for better understanding of the mechanisms behind human health and disease, is investment across this entire continuum, from the biomedical research space and basic science through to validating and qualifying models that represent human biology and how that biology can be perturbed and lead to disease, and then leading to adoption and implementation, not just within the US, but worldwide. A very, very quick overview of a new program that's just been launched by the NIH Common Fund that I hope many of you have heard of. It's called Complement Animal Research and Experimentation, or complementary, really intended to address the first part of this continuum in terms of providing funding for biomedical research and platforms that are fundamentally rooted in human biology. And we just released a notice of intent to publish a funding opportunity. That funding opportunity will come out in October. I would encourage you to go to this website and sign up for the listserv so you don't miss future announcements. But the way that this complementary program is going to be structured is a comprehensive center model, which will have technology development centers and data hubs and community engagement and training. But today I'm really here to focus on this validation piece. So we will have a validation and qualification network as part of complementary, and this is really building off of some of the tremendous successes that ICFAM has demonstrated over the last several decades, and ICFAM is a key partner in this effort. So who are ICFAM? For those of you who may not know, we are the U.S. validation body, so the Interagency Coordinating Center for the Validation of Alternative Methods. We have 18 different federal agencies, and we are co-chaired by representatives from the FDA and the Department of Defense. And about six and a half years ago now, we published our strategic roadmap, which was really intended to be an evergreen document. So very relevant still today. It describes these three key processes that have to be established to really facilitate effective and efficient fit for purpose validation approaches and ensuring that methods are being developed to meet the needs of our end users. And so that is really represented by the figure on the front cover here. So having constant communication between end users, validation bodies, and method developers. But as we were developing this roadmap, it became very clear to us that there was really no such thing as a one-size-fits-all approach to this process. And here's an example of just 2 out of the 18 federal agencies that belong to ICFAM. Admittedly, the 2 sort of big regulatory players in this space, FDA and EPA. But there's no such thing as validating a method for FDA or for EPA, because within each of these organizations, you have different centers and different offices that deal with everything from drugs to food additives to personal care product ingredients to industrial chemicals to pesticides, et cetera. So to consider this, very complex landscape of federal agencies and the offices and the centers therein, and the diversity of chemicals and products that they regulate, we decided to embark on a deeper dive into validation, qualification, and to ensure or facilitate regulatory acceptance of new approach methodologies, or NAMs. So we formed the ICFAM validation work group, and for the last 2 years, we worked on developing this guidance document, which is really a follow-up to the roadmap. This was published earlier this year in March, and it's a real detailed how-to guide on how to consider some of these key concepts, again, represented on the front cover. So you'll see a theme here. If nobody gets past the front cover of our documents, they'll at least get the major takeaways that we want people to understand. So I'm going to go through some of these key concepts in more detail for the rest of the presentation. So again, here's another figure. Here's another version of that figure. So the key concepts within this guidance document are things like fitness for purpose, or the context of use of the method, understanding and demonstrating biological relevance of the method, data integrity, technical characterization, information transparency, and independent review. I would encourage all of you to go and read this in detail. There's a lot of great information in there that I won't be able to cover or do justice to today, but I'm going to try. So the first key concept that people need to consider when trying to validate a method or to develop a method that is intended for ultimate implementation and regulatory use is, what is the purpose of that method, also known as the context of use. So a lot of the content in the ICFAM guidance document builds off of or is borrowed from a recent publication that we put together with some of our NGO colleagues and some of our international colleagues from the European Center for Validation of Alternative Methods, or OECD as well. And these considerations are things like, what regulatory statutes are the data from the NAM intended to comply with? Is the NAM intended to be used on its own or as part of an integrated strategy? How is it intended to be used? And what regulatory endpoints is it intended to address? So is it things like screening and prioritization, all the way down to, deriving a point of departure for quantitative risk assessment? So this is the first key consideration. The second key consideration is biological relevance, also sometimes called mechanistic relevance. So demonstrating similarities between the biology that's being measured by the method or the test system or the testing strategy and the biology of the species of interest. In this case, I put human in parentheses because much of the time we're talking about human health endpoints, but this is equally relevant and applicable to ecological assessments and methods that are designed to predict toxicity to other species, other taxa. Here's a couple of examples within the ICFAM validation report where that approach, biological and mechanistic relevance, was really the driving force to demonstrating that NAMs are applicable and can be used in regulatory decision making. And there are many more of these examples, But these are the ones that we really wanted to highlight because they have associated regulatory guidance documents and applications within regulatory decision frameworks, either by the US EPA or internationally at an OECD level. So skin sensitization has, a very well-developed adverse outcome pathway. Methods were mapped to molecular initiating events or key cellular events, and then combined in defined approaches that were then codified in an internationally harmonized OECD test guideline that is accepted as a full replacement to historical animal tests that have traditionally been used. Endocrine disruption, there's different pathway models, both integrating in vitro data or using quantitative structure activity relationship models that are accepted as part of the endocrine disruptor screening program, sort of tiered testing strategy. Developmental neurotoxicity, there's an in vitro battery where there's an OECD guidance document on that, and various authorities are now starting to apply that battery to screening compounds. Inhalation toxicity, this was a case study that was reviewed by the EPA's Scientific Advisory Board and used to waive the requirement for a in vivo study for chlorothalonil. This has also been published under the OECD. So there's a lot of additional detail in the guidance document on these specific examples. Another example that at the time of publishing or drafting the validation guidance document wasn't available yet, but has since been codified into regulatory guidance is eye irritation and corrosion. So there's been a tremendous amount of work to assess different approach methodologies based on their relevance to human biology and the mechanisms of eye irritation and corrosion in humans, that then resulted in this new chemicals program decision framework from the US EPA, OPPT, for hazard identification of eye irritation and corrosion. And I don't expect you to read this. It's kind of a complicated decision tree, but the take home here is that they are highly prioritizing data from human cell-based or human tissue-based test methods that have been demonstrated to be reproducible and relevant to eye irritation. And that sort of biological relevance piece was really the driving factor in releasing this regulatory guidance and this framework. The next aspect within this confidence framework is technical characterization. And Dr. McCulloch, who will speak after me, is going to do a real deep dive into many of the finer aspects of technical characterization and some of the really important considerations in that space. So I won't belabor some of those points. I do just want to highlight this one sub bullet under technical characterization, which is accuracy. So this is often the linchpin of a validation study. So you have a new approach and you're trying to demonstrate that it performs as well or better than an existing historical reference test method. And so traditional animal test methods are usually the benchmark that's used for comparison, but it's really, really important to consider that those animal data are at best a surrogate for the endpoint that we're really trying to predict, which is a human health endpoint. And so taking a more transparent look at that historical animal data that's being used as a benchmark and understanding the relevance of that system to predict human health effects, but also the variability of that animal data and characterizing and quantifying that so that you can put a confidence interval around that endpoint and set appropriate expectations for the performance of a new approach method that might be proposed to replace that. And so I'm going to talk through an example of where we've tried to incorporate that concept of reference data variability and how it is used when both benchmarking predictive performance of new approach methods, but also in a sort of data-driven, transparent way to put confidence intervals around those predictions. And this is an example from an international collaborative project where we essentially crowdsourced across computational chemistry groups across the world models to predict endpoints like endocrine disruption, or in this case, I'm going to talk about acute systemic toxicity, so the CATMOS project. And it's also worth noting that these other global projects that predict things like estrogenicity and anti-androgenicity have been validated by the EPA, are accepted as part of a weight of evidence strategy within this EDSP program and by under REACH by the European Chemicals Authority. But CATMOS was the most recent project that we did, and all of these are available under the Opera, so the open QSAR application that's available through the NIHS GitHub. And here, we, as with all these other projects, we curated a large set of training data that we gave to all these different groups around the world. They built their computational models, gave them back to us. We assessed them both quantitatively and qualitatively, and then pulled the best models together to create ensemble or consensus predictions that provide broad coverage across the entire chemical universe with defined structures and are very, very robust and reliable models. At the same time, we noted that we had a subset of compounds, over 1,000 compounds, that had replicate animal studies. And so that allowed us to use a statistical bootstrapping approach to look at the variability in those animal data that were being used as a benchmark for comparison, and derive a 95% confidence interval that not only we could apply to the predictive performance assessment, but that we could also apply to the predictions that came out of the QSAR app. And so then we worked with the US EPA to assess the ability of this AI-based computational predictive model, so this fully in silico QSAR tool, to predict the LD50. And we looked retrospectively at how the LD50 had been used in risk assessments over the last several decades, and looked across 177 different pesticide active ingredients that had existing LD50 data and asked how well did CATMOS predict those LD50s that were used as the basis for risk assessments. And basically what we found is that it was extremely reliable in predicting non-toxic substances, which makes a lot of sense because that was the vast majority of the training data that went into training this computational model. And so if you consider, for example, EPA categories 3 and 4 together, so these more non-toxic substances, chemicals that were predicted by CATMOS to be in this range were consistent with the in vivo category assignments 97% of the time. And so we're hoping that this was recently published in this Bishop et al. Paper, which EPA co-authored with us, and we're hoping that this will form the basis for a waiver guidance where if a compound is predicted to be non-toxic by the model, then that will waive the requirement to do the animal test. The next key concept in this framework is data integrity and information transparency. So this is a huge aspect that's under consideration under things like the OECD test guidelines program as well. So there's a real balance to be struck here between maintaining like intellectual property and ownership of a method by a company or a method developer, and being able to independently assess the validity of that data and those predictions, and to be able to reproduce the data and the predictions coming out of a model, whether it's an in vitro model, an in silico model, or some combination of those strategies. So that's a really, really important aspect of the process. Another important aspect of information transparency is widespread accessibility. So the defined approaches to skin sensitization that I described briefly in a previous slide that are codified in this OECD test guideline are, for the moment, pretty simple rule-based approaches, but they're still subject to error and just mistakes and how they can be applied. And so to facilitate more widespread uptake and to sort of remove the potential for human error, we developed what's called the DAS app, so the Defined Approach for Skin Sensitization app, that's available through the NIHS website, that allows users to just upload their own data or pull in data from our integrated chemical environment and automatically apply the defined approaches that are in the OECD test guideline. Okay, the last aspect of the key concepts of this framework is the concept of independent review. And this is a really important part of the confidence building process, but it could look different depending on the intended context of use of the method. In some cases, having a series of peer-reviewed publications may be sufficient for an agency to consider putting a method into their decision framework. But if you're trying to get all the way to, for example, an OECD test guideline, that typically needs a formal, independently coordinated peer review process. So one example of a validation study that NICEDM is supporting right now is this transferability and validation study of a human thyroid microtissue assay. So this is a 3D assay that was developed by the US EPA, consisting of primary human thyroid cells that assesses thyroid hormone inhibition, which is a key hub in the thyroid AOP network. And this study is being coordinated by NICEDM, but we have multiple different participating labs, and this will be peer reviewed externally in a way that is coordinated independently by our policy office. So I'm excited about the outcome of this study. It's going really well so far. I want to kind of dig in a little bit now on some, I think, really helpful guidance, specifically in the area of in silico and AI-based applications. So among all of our federal agencies, I would say that FDA is really the leader in this space in terms of how they are considering credibility of computational modeling and simulation and issuing guidance around its use in healthcare applications. So in 2021, they published the AI ML-based software as a medical device, so the SAMD action plan. And that was subsequently followed up by much more detailed guidance. So at the end of 2023, they finalized this guidance around assessing the credibility of computational modeling and simulation, which really builds on the sort of FDA recognized standard, the ASME VNV 40, and really kind of brings in more sort of non-traditional sources of evidence and kind of this like standard approach to validation that's described in the VNV 40, which is like well-controlled bench testing. But this guidance from FDA brings in things like clinical studies, population-based validation results, et cetera, which really might be more aligned with the model's context of use. And then more recently, in March of this year, they put out a white paper discussing how several of their centers, so their centers for biologics, for drugs, for medical devices, and combination products, are aligning their efforts to advance the responsible use of artificial intelligence in the healthcare space. They have 4 areas of focus: fostering collaboration, advancing the development of regulatory approaches, harmonized standards, and supporting research in this space. And then even more recently than that, in June of this year, I was fortunate enough to be part of a team of people from across industry, academia, and government, in particular led by Tina Morrison, who directs the Office of Regulatory Science and Innovation, and coordinated and supported by the Reagan-Udall Foundation for the FDA, that is really a white paper that kind of brings the value proposition for using in silico and AI-based methodologies to companies. So it's aimed at decision makers advocating the use of in silico methods across all industries regulated by the FDA. And there's a number of really great examples in here. So liver safety assessment based on quantitative systems toxicology and PBPK modeling, personalized medical devices in silico patients using finite element analysis, all kinds of great examples. And I'm running out of time, but I'm just going to quickly highlight figure 4 from the report, which kind of gives an overview of the use of modeling and simulation across the FDA, organized by discipline in the rows, and then application area in the outer columns, and FDA center in the inner columns, and how they're color coded. So you can see that AI and machine learning and big data driven models, there's a lot of opportunity, I think, to use that in various application areas across most centers in the FDA. And then the very last thing I'll highlight is, having a platform to support all of our validation efforts is really, really important for NICEDM and for ICFAM. And that platform is the Integrated Chemical Environment. It provides access to very robust data sets of reference data, of new approach method data, computational toxicology workflows. And you can just see a couple of screenshots of things that you can do in ICE in terms of exploring high throughput screening data, looking at the presence of chemicals in consumer products, running virtual animal studies or human studies, et cetera. So I encourage you to check that out. This is our new group photo. It was just taken a couple weeks ago. I am super privileged to work with all these amazing people. And I will stop now and turn it over to Sean.
Hey, Sean, are you ready to go share your slides? And I see there's a lot of questions in the chat. We'll go ahead and start addressing some of those. And then the ones that we can, we'll hold to the end and share as the group.
Getting the share going right now. All right. Well, thanks everyone. Great talk, Nicole. Looking forward to addressing and building on some of what you raised. So we're going to focus, I'm going to get a little bit down in the weeds on some concepts in vitro systems and assays. We're going to focus on respiratory toxicity because it's a little less developed than a lot of other systems and assays out there in general. So it gives us a little bit to think about, but a lot of this also applies more generally across tissues and assays for other types of toxicity. So I'm a senior respiratory scientist at RTI International, where we do a lot of in vitro modeling, both for basic science and commercial testing. And so we got a lot of ground to cover, so I am gonna get moving. So a lot of folks on here are gonna be familiar with sort of our overarching problem, which is we got tons and tons of chemicals with very little data. We have a small number of chemicals with a lot, but generally we have a lot with a little, and then we need to increase the human relevance of what we've got. And a lot of this is compounded by the factors that we've got listed here. That's mostly a consideration for screening. It's important to differentiate between applications in screening and then applications in testing, where we have maybe one or two discrete compounds and or scenarios that we're really going to dig deep in and try to get a little bit more human relevance than the existing in vivo animal models will allow. So to get right into the respiratory tract, so a lot of respiratory toxicity modeling has focused on the airway epithelium, and that's for a number of reasons. One of those reasons is it's the outermost layer of your body that interacts with the environment that you breathe in. So it's going to kind of have the first take at whatever you're breathing in, and it functions as the barrier between your body and the outside world. So it's a physical barrier. It also has something called mucociliary clearance, which is a cool word, always fun to say when you have an audience. But basically stuff gets caught up in the mucus that it secretes, and it has cilia that'll just roll it up out. And sorry, if it's lunchtime for everyone, you either cough it up or you swallow it down. And then the respiratory epithelium also coordinates the local immune response by secreting pro-inflammatory cytokines and plays a role in concert with other cell types in the area of orchestrating remodeling after injury. So we have a couple of nice advantages in the in vitro respiratory tox world. So if we now think about that tissue section that I just had up there, and only think about these dark purple cells at the top, which are the, let me get a pointer here, laser pointer. So these dark pink guys are the respiratory epithelium. We've got goblet cells here. We've got cilia up here on ciliated cells. And then we've got these little cone-shaped guys down here are basal cells. And if we take respiratory epithelial cells, which we can isolate directly from donors, so these are primary cells, we can create these epithelial-only air-liquid interface differentiated cultures, and they very nicely represent that architecture from the in vivo section. We can also, if we sort of zoom out and take a bigger look here, we can start incorporating this big pink mass that's a lot bigger than this dark pink mass up here. So this is stromal fibroblasts. This is connective tissue. We've got collagen. We've got a number of other cell types, but mostly this is fibroblasts. And fibroblasts have a terrible publicist. I do my best, but I'm only one person. And so I'm going to tell you a little bit about fibroblasts today and why they're important and why they're important for complexity here. But these cultures can be generated in-house. My lab has done this at EPA and RTI, and these are also commercially available from different tissue providers. And so this is an image here where you can see the epithelium on top of a porous membrane, and then underneath here you have these lung fibroblasts. So in vitro respiratory models have been integrated into a couple of decision-making or regulatory decision-making applications. The sort of first and leading one was this case study for chlorothalonil. This leveraged an epithelial-only system from 5 donors, and this work was done. Data were collected, I believe, between 2016 and 2017, and then there was a final report in 2018. And then if you fast forward about 6 or so years in the future, being now, there was a more recent in early 2023, there was a Toxic Substances Control Act, or TSCA, test order released for an inhalable PFAS gas that was the first to specifically require the use of an in vitro respiratory tract epithelial cell system for toxicity assessment. And in this case, this work is actually being done in an epithelial and fibroblast co-culture. So just in those 6 years or so since the chlorothalonil study, we've had a pretty good advancement in the consideration of using these multicellular systems to increase the biological relevance of these systems, as Nicole was talking about. So since we're not just talking about biology today, we're talking about validation, I want to sort of take a couple of excerpts from the validation principles that are highlighted in OECD Guidance Document 34 that are going to be kind of cross threads through what we're talking about today. So we want to be thinking about how our in vitro systems and assays are related to the endpoints and the test methods and the biology and toxicity that we want to model. We want to be able to understand and report and characterize limitations as well as applications. We want to be able to understand and demonstrate intra-test variability, repeatability, reproducibility within and across laboratories. And we want to be able to evaluate this test method relative to existing toxicity data and ideally the in vivo scenario. So really, if we take then one step back from that, we have to think, are we ready for that? And the short answer is we're not there yet. So instead of just focusing on validation, what we can focus on today in this context is characterization, because we really need to have a much better understanding of how these systems and assays work and how we can use them. And so without addressing these, we really can't take that step towards validation. So we're going to talk today about integrating some in vivo relevance and how that allows us to capture biological effects and ensure representation of our toxicity of interest, thinking about providing context for applications and limitations of approaches, and then identifying factors that may impact the variability, repeatability, and reproducibility of some of your in vitro assays. So part one, let's talk some biology. So basically, we start with a question in the respiratory field of which system is right. And so this is one of those things where you can have, you can sit down and you can have a lot of philosophical discussions and opine for probably ever with different people and get a lot of different opinions on where are we, what should we be doing, how simple, how complex do we need to be. I would make the argument that even though we may be looking for an event that the epithelium is involved in the toxicity, the other cell types within the tissue, and I'll show you that in a second, are involved in how those epithelial cells respond. So we're going to be kind of thinking about things in that context. But several years ago, my lab asked this question of how complex do we have to be? If we integrate fibroblasts, are we going to see something different? And so we came up with what we call the transepithelial exposure model. Anybody that knows my lab knows we have fun acronyms and kitschy names for things. And so what we've got here is the bronchial epithelial cells and then underlying lung fibroblasts. And we separate these. This is an indirect co-culture. Happy to discuss why that, the pros and cons of that. But basically, it allows us to represent this tissue more or less, and we can capture the effects of direct exposures, which is material interacting with the epithelial cells, and transepithelial exposures, or if you have an exposure up in this epithelial compartment, then we'd be looking at what's going on in the fibroblasts. And we design this to be very simple. Its original iteration and the first set of data that I'll show you were from a cell line based model. And then since it's evolved and now we have donor matched primary cell based systems that are at ALI, where we can do all sorts of exposures with them. So we can have this co-culture system. We can expose it to a test article. In the first data set, it'll be diesel exhaust particulates. And then we can sample here to look at the whole culture response. And then we can also take it apart. And then we can isolate the epithelial cells and fibroblasts separately for parallel analysis. And so I'll give you a very abridged version that this paper was published in Toxicity in 2020. It's Faber et al. If you want to look it up and sort of get into it. But if you look at some of the expressed genes here, and in this case, we'll start out with a transcription factor. So we see in both the bronchial epithelial cells and the lung fibroblasts, again, these were exposed in a single system. We see the induction of NRF2, surprisingly, at least to us at the time, with very similar kinetics, suggesting that even though in this case we didn't see a change in barrier permeability, suggesting that it's not just the particulates and associated things going through, but that there's some other mediator there and that the fibroblasts are responding very quickly despite a distance and dilution between those two cell types. We also see an increase in the expression of several oxidative stress responsive proteins in fibroblasts in a much greater sense than the epithelial cells. So we've got heme oxygenase 1 and QO1, and then PTGS2, more commonly known as COX2, which is actually very highly induced in the fibroblasts, but not at all in the epithelial cells. We're also seeing the persistence of these upregulatory events into the 24 hours of exposure time period. This study didn't look past that, but it does suggest implications for secondary exposures or longer-term outcomes. More studies to come, I suppose. Also in this study, we did what I think at least is a pretty cool experiment. So we took this system, and one of the advantages of having this insert is we can take the epithelial cells out. We can specifically target the fibroblasts. In this case, we did it with the antioxidant N-acetylcysteine, and we can incubate it, and then we can wash it out, and we can put the system back together. And then we can conduct our exposures. And so when we did this, we expected to see a change in or mitigation of the induction of oxidative stress responsive targets in the fibroblasts. And that happened, and that made sense. Great, box checked. What we expected less, and we're much more excited to see, is that when you treat the fibroblasts with the antioxidant, again, it's flushed out of the system and then expose the epithelial cells, it's also mitigating the effect of the epithelial cells. And so what this is telling us is that the redox state of the fibroblast is influencing the way that the adjacent epithelial cells are going to respond to a stimulus, even though the epithelial cells don't see that antioxidant. So the core point there is that the fibroblasts are influencing the epithelial cells. And this is not new. This has been shown over and over again, mostly in the cancer literature with tumor stromal interactions. Okay, so now we're going to move a little bit farther down the respiratory tract. So this was another model that was constructed by a very talented graduate student in my lab, Eva Vitucci, who has since graduated. And this is focusing on the alveolar microvascular interface. So this is the alveolar capillary region exposure, or ACRE model. And so we've got alveolar epithelial cells, interstitial fibroblasts, and lung microvascular endothelial cells here. They form a nice Z01 mediated tight junction. And they also have adherence junctions. So we've got a good barrier here, which has a good tier value. Similar to the team model I just showed you, we can conduct this exposure. We can take the system apart. In this case, we removed the fibroblasts. We were focusing on the epithelial cells and endothelial cells, and we can conduct that parallel cell type specific analysis. And what we did in this study is subject those samples to RNA sequencing. And what we saw here is that the epithelial cell and endothelial cell responses at 6 hours were notably different. And that continued on as we looked out to a 24-hour exposure, where we see what appears to be more or less a resolution in the epithelial cells, but you have a continued effect that increases in the endothelial cells. So this tells us a couple of things. A, that time is important. So we should always be thinking about the kinetics of our response and doing time courses. But also it tells us that if we had just used these epithelial cells, we would have missed whatever's going on in the endothelial cells. And this is a little bit farther in the weeds, but it was such an elegant study done by the student. So I would really want to show it because at least I think it's cool, and maybe I'm just a big nerd. But so we generated a second version of this model. So we've got the model we've been talking about with 3 cell types, and then a version that just has the epithelial cells and the fibroblasts, no endothelial cells. We did these exposures. We looked for release of the IL-8 or secretion of IL-8 protein into the medium. And when we have the full model, we see a significant increase in IL-8 secretion. But when we don't have the endothelial cells, we don't see anything there. And this was supported by now when we isolate these cells and isolate the RNA and do qPCR for IL-8 in these are HuLEC cells, the endothelial cells, we see a significant increase in the HuLEC cells, but not in the epithelial cells. So that is supporting that the endothelial cells here are critical for the secretion of IL-8. And then if we really want to get mechanistic, which is of course relevant to integrating data into AOPs, so we had done some blotting to see that NRF2 is activated in the endothelial cells, and MAP kinases, specifically ERK and P38, were activated in the epithelial cells. And so we've got an increase in IL-8 RNA this is in the endothelial cells for DEP, which we've seen before. If we specifically knock down NRF2 in the endothelial cells, we mitigate that effect. So that's telling us that endothelial NRF2 is required for this event. And then if we take it sort of one step up the chain and ask the question, well, is the MAP kinase activation in the epithelial cells required for the activation of NRF2 in endothelial cells? Turns out the answer there is yes. And so then the next question, which is really the exciting one, is the activation of MAP kinases in epithelial cells required for the induction of IL-8 mRNA in the endothelial cells? And again, the answer is yes. So that ties this nice little mechanism down where we have the exposure occurring in the epithelial cells. We're activating one set of signaling pathways. And then we've got, I really need to put a black box and a lightning bolt here to have like the international accepted version of just we don't know what's going on in the middle. But that leads to the activation of NRF2 and the transcription of IL-8 and then translation and secretion of IL-8 protein. So obviously in this case, if we didn't have the endothelial cells in the system, we would have missed a lot. Now, if we take a quick pivot and we're going back to our bronchial model now with primary cells to think about. So we want to be thinking about how do we reflect a diverse population when we do toxicity testing? Obviously, humans are diverse. We have a range of responses to things, and we want to make sure that as we develop new toxicity testing methods, that those methods are equally protective across our diverse population. And so this is pretty challenging with inbred animal strains or isogenic cell lines, which have minimal genetic variability and likely even less epigenetic variability. But when we get primary cells, we can get them from a diverse human population. And thus, there's a potential there for those cultures to represent that diverse population in our toxicity testing. And so we did this with donor match co-cultures with bronchoepithelial cells and fibroblasts that were derived from 14 individual donors. And so all these donors are being treated individually. We're not pooling them here. And so even before we get a toxicant involved, you can look at the architecture of the epithelial layer, and we've got even just sort of differences in organization between 58P and 40I. Then we've got clear differences in thickness of the epithelial layer to 50Q, and then differences in the distribution of different cell types that make up this layer. In 48M, what this is showing is that we've got a bunch of goblet cells compared to other cells that we see in these other sections here. And so if we then take that and we took those cultures and did a concentration response experiment to acrolein, which is a sort of ubiquitous reactive gas. And we looked at the effect of variability on a number of common endpoints that were relevant to, and this is key, relevant to the known outcomes in vivo studies for acrolein and incidental human exposures. And so if we look at just overt cytotoxicity, we see a range of effectively no effect to all the way dead. Barrier integrity measured by tier. So we see a lot of range of variation here. And then things like IL-8 secretion, a lot more variation here. And these are just sort of representative endpoints we're working on writing this paper up. We've got a whole lot more. Some are much more variable than others. Some of these aren't variable at all. And so it's kind of an interesting story that we're trying to wade through. And so now we've thought about inter-individual variability at the donor level, but sort of going back to the cellular level and taking an extension off of that alveolar model that we talked about a minute ago. And so now in this study, we also collected, we can take the system apart and we can isolate epithelial cells and fibroblasts, isolate RNA, submitted it for RNA sequencing. And that was thanks to some nice coordination with Nicole and Dr. Julie Foley at NIHS, and then some analysis by Dr. Martin at NIHS, who's now ICF. And so we asked the question, are alternatively regulated gene signatures driven more by donor or by the concentration of the toxicant? And in the epithelial cells, the principal component analysis doesn't show any clustering by donor, but it does show pretty neat clusters by dose. And so you can see with these colors here, I would have put ovals over them, but they overlap too much. So sorry about that. But you can see we've got some nice clustering here. Not too surprising, a little surprising we didn't see a donor effect. But then when we get into the fibroblasts, we see clustering exclusively by donor and effectively not by dose. And so this was really a lot of head scratching, and we're still thinking our way through this and working on the analysis of these data. But what it tells us is not only do we have, and we've got differential expression analysis showing that the genes are different, but so we've got differentially expressed genes, but also like the core biological mechanism by which the cells respond to these exposures is different. So thus making the argument that it's important to capture this in the experimental system. And so what this really does is it tells us we need to drive the biological relevance of these test systems, and that we should probably really think about moving away from epithelial only systems, even though, yes, they're simple, everybody's familiar with them, but we're seeing a lot more data that indicate that these other cell types are relevant, even to the epithelial response. We also have to consider inter-individual variability, not only to inform sample size, but also to understand the variability within tests, the repeatability and reproducibility of these studies across laboratories and occurrences. And so also we need to consider this as a variable in the range of variation in performance in response to reference agents and then interpretation of in vivo human data. All right, dosing methods. So ALI cultures are, well, trying. There we go. So the main benefit of these ALI cultures is that they have air on the surface of them, like the lining of the respiratory tract. So that's a benefit, but it raises a challenge to do these exposures, because maintaining ALI conditions during exposures is very technically challenging, typically requires specialized apparatus and specialized expertise. So a lot of these exposures are done by something called liquid application, where you put your test article in some kind of liquid solvent, and then you put it on top of the cells for however long, and you do your assays. Historically, this is done with an anecdotal volume, typically between 10 and 30 microliters. At least the published literature does not have any indication of anybody that's done any work to justify those doses. And so we asked that question a number of years ago and have been working on it ever since. And so we started this, and these are 6.5 millimeter inserts, which is a typical format used for a lot of testing applications. And when we put this volume, this is trypan blue, which is a cell exclusion dye, in 0.9 saline at 10 microliters, 30, and 50. At 0 hours, 10 microliters doesn't even cover the culture completely, 30 does, 50 does. And now if we fast forward 24 hours, 10 microliters, definitely not. 30 microliters, we're seeing a gap in the middle, 50 still covered. And we see these little mucus balls forming here. This is something that occurs across a number of buffers. We'll talk about it in a second. But the reason why this is important is because when you get these differential coverage events, you're really now causing a gradient in test article concentration, ion concentrations, oxygen, nutrients, and you're not getting an even event. So you may very well be, when you harvest these cells, especially if you're doing something like transcriptomics or protein analysis, you're going to get a continuum that may just sort of average out to 0, but we don't know. So it's sort of something that you want to keep in mind as you're thinking about how we do these exposures. And so we kept thinking about it. And then this was work done by Dr. Aaron Huber, who's an investigator in my group at RTI. And so we took these cultures and put a distribution of 10 micron green fluorescent polystyrene beads on them and just sort of tracked it over time. And the cells sort of roll this up into what is, for lack of a better term, a green booger. And then I've got to exit my pointer. And then they more or less roll it around the culture and in real time. And it's actually pretty interesting because it's relevant because that's what your mucociliary escalator is going to do in vivo, but it also creates a problem in terms of in vitro, the use of in vitro systems because of exposure and sequestration of your toxicant. So it adds a couple of challenges. So we asked the question in an intentional study where we basically wanted to figure out what's the effect of liquid application on an in vitro ALI system. So instead of using anecdotal volumes, we collected some data on, basically what volume is going to keep coverage at 24 hours. We had to use a larger format here, 12 millimeter inserts, to accommodate our endpoints. So note the volumes are different, but these all scale down. We also then selected our vehicle, which it was ALI medium, based on it's going to be an appropriate pH and not result in starvation of the cells during the exposure, compared to 0.9 saline and cell culture grade water, which are commonly used. Let me get my. But what's often not appreciated is that these guys are very low pH for biological systems. So that's something to be considered here. So we took our co-cultures and we maintained some at air-liquid interface, and we put liquid on some for 6 or 24 hours. We did some endpoint assays, then we separated, isolated cells, and did RNA sequencing. And we did both, but we'll focus on epithelial cells today. And what we saw by 6 hours is that we had over 4,000 significantly alternatively regulated genes, which increased to over 10,000 by 24 hours. And then we see sort of this progressive effect into the 24-hour alternatively regulated gene number here. And if we did IPA analysis on these, we see that we've got pathways related to tumor microenvironment, fibrosis, hypoxic signaling, injury, wound healing, these sorts of things. Never mind that we've got things like hepatic here. If you break it down, these are being driven by things like placental growth factor, IL-8, TNF alpha, VEGF. So it's it says hepatic, but it's more of a just general fibrosis, injury, and repair pathway. So then we sort of took the scientific validation route and wanted to say, okay, well, we see all these. Do we see these effects at the pathway level? Turns out, yes, HIF1 alpha is stabilized, meaning a hypoxic activation. We see a very strong activation of hyperphosphorylation of P38. Same with ERK1, but not ERK2. And then this also led to our identification of this hypersecretion of cytokines like IL-8, TNF alpha, and then growth factors, VEGF, and placental growth factor. So at the end of the day, what we take away from this is that by putting liquid on these cultures, we're basically causing an atypical state that's reflective of a number of respiratory disease states and indicative of a pre-epithelial to mesenchymal transition phenotype, or pre-EMT, which is one of the early steps in carcinogenesis. And this was supported by our observation that these cultures that are submerged start having a lower barrier integrity that leads to increased permeability. So again, you're pushing the cells away from a typical epithelial phenotype into something else, typically squamous cells. And that here's just a graphical representation. We've got a bunch of pathways that get activated. We're secreting growth factors and cytokines and breaking down our barrier integrity just by the dosing method alone. And so basically, and I'll try to shorten this up for time, we want to understand the effects of our dosing method so we can extrapolate that into how we interpret the limitations and applications and interpretations of our study data. So if we move on, we'll do a real quick but deep dive into one method that's very common in the respiratory space. So this is TEER, trans-epithelial electrical resistance. It measures epithelial barrier integrity. So again, we're looking at epithelial cells, which line your respiratory tract. When they get injured, their integrity goes down, so their permeability goes up. This can be measured by measuring the resistance across this cell layer. There are a number of different ways to do this, and I'm just showing a very brief smattering of data from a characterization done by Dr. Aaron Huber. So there are a bunch of different electrodes you can use to measure this, and even the equipment that you select for this can impact your outcome. So this is just measuring blank resistance of inserts. The endome chamber, so these chambers here that fit the insert, gives you a much lower and significantly different value from these STX4 chopsticks, and then again, different from STX2 chopsticks. Not saying one of these is right or wrong, but it's important to understand that they're different and understand and report what you're using and how that may impact your outcomes. Also, there are preceding, so this is before you even put the cells in place, there are considerations there that you should think about, such as, does the manufacturer of the insert affect the resistance of the insert? Answer there is sometimes yes and sometimes no. These are looking at 3 manufacturers in red. So we've got a significant difference between 2 of them, but not a different 2. And then in cases like ALI culture, you typically coat your inserts, but that doesn't seem to affect the TEER. And these effect of manufacture can be just due to pore size or pore density. Again, not right or wrong, it's just different, and we need to understand that. And then the way the assay is actually conducted varies significantly across laboratories. And so a lot of folks will wash the apical surface of their culture and then leave that wash buffer in place for the measurement. And what we can see if we compare to using ALI medium in both compartments, which is what's recommended by the manufacturer, we see significant variation of, basically we lose a lot of TEER when we do that using the wash buffer in the apical compartment. If we then do this where we have ALI medium in both compartments, but have washed with all those same buffers before doing this, we mitigate a lot of those effects, but we still do see some significant effects here of some of these treatments, including things like 0.9 saline, which is one of the most commonly used buffers in the ALI culture systems. And so next, positive control selection. I'll be really brief here, but oftentimes things like detergents like Triton X or SDS are used as positive controls for tear, and they will reduce your tear, but it's because they're permeabilizing and killing your cells, not because they're reducing barrier integrity. So while cytotoxicity can increase barrier integrity, we need to be conscious that that's not the only thing that we want to capture. And so we tested EGTA and found it to be a very nice and reliable positive control for reducing tear without killing the cells. And lastly, maintenance is really important. And here is a video of 2 different endome chambers, and we might have a connectivity issue here. But the short of it is, basically, we were asked by some colleagues to test a chamber, and the variability in the measurements of this chamber were extremely high relative to where they should be. And so we were able to, are we going to see it now? All right. So ultimately, what we figured out was that there was a lot of corrosion in the electrodes, shown here in a Q-tip. This is what the bottom electrode should look like versus here. And we also found a lot of corrosion on the cap electrode. And so there were some other things going on here, but just as a very, it's not only the biological stuff that we have to keep in mind, we have to make sure we're maintaining our equipment properly. So quick summary, basically, we have to characterize our systems and assays to make sure that we're ready to go forward to validation, or we're kind of setting ourselves up to fail and spend a lot of money doing so. A lot of these methods and systems remain poorly characterized, and doing this characterization allows us to align systems and methods to the in vivo biology and the toxicity endpoints that we're trying to mimic in vitro. It also allows us to understand and disclose the applicability and limitations of different systems and assays, and informs how we develop standardized protocols, again, to support validation efforts and harmonization across laboratories. Had a lot of colleagues on this that I'd like to thank for time. I'll keep it brief. And so if we have any questions, happy to take them now.
So first off, thank you guys for the great presentations. You both really know how to pack in a lot of information. Those slides and the recording will be available, probably be about a week, and folks will get an email. So we had a question about, for folks new to this, how are they going to, where should they go to get started? And Nicole answered in the chat, referring to the NICEDM website, which is a really great resource. But when folks are starting to think about getting these integrated into their workflows, or bringing in either a computational or an in vitro system, what are some of the things they should be considering as they approach this? And I'll let Nicole maybe answer first on the computational side and bigger, and then Sean maybe weigh in on some of the in vitro.
Yeah, thanks, Shannon. So, I mean, a big thing is, accessibility of the tools, so you're looking at a computational resource. We, with ICE, as I briefly alluded to, we're trying to sort of democratize access to computational toxicology workflows and tools that were previously just the domain of subject matter experts. So I'm thinking about things like IVIVE, in vitro to in vivo extrapolation, being able to run PBPK models, do structure-based read across, et cetera. And so we have spent a lot of time and effort putting out training resources that are associated with those tools. So help videos and standard operating procedures and things like that. But also kind of trying to decide what method or what technology is applicable to your chemistry of interest or to your endpoint of interest, and what's going to be accepted or at least considered by the regulatory authority that operates in the space that you're also operating in is another key question, right? And so we actually have a new resource that just got awarded significant internal funding to stand up, called CAMERA. It's going to be the Collection of Alternative Methods for Regulatory Application, which is going to be a database that people are going to be able to come to and ask the question of, okay, I want, a in vitro eye irritation method. Which of these 5 that I'll fall under this OECD test guideline is appropriate for my chemistry, and use a data-driven approach to assess applicability domain and understand which agencies will accept it for which context of use, et cetera. So yeah, I'll turn it over to Sean.
Yeah, all that, and making sure that you're on the in vitro side, especially, always thinking everything's respiratory, right? So we got to think of what's the real world exposure scenario that you're trying to model. Is it somebody using a pesticide applicator? Is it somebody that's, the next farm over? Is it somebody in an industrial or personal care setting? And how is that going to influence not only the way that you expose your test system to the test article, but the concentrations that you're going to use in doing so? I think we also need to approach this with an open mind to have a wide range of concentrations to test, thinking in the context that we don't know what the translation between cells on an insert or in a dish, and in the case of an ALI system, how does that, what's the quantitative extrapolation from that to the respiratory tract? You can do some MPPD or CFD modeling, but that only captures the sort of the deposition of the exposure. It's not capturing any biology that may be present or absent or at play here. That's going to lead to differences in what your concentration response is going to be in vitro and what it would be in vivo, whether that's in an animal or a human. The second, what Nicole say, you really need to think of what question are you asking and how are you doing everything that you can to provide as much context in asking that question as you can, and then follow up with making sure that you're reporting as much as possible, because it all makes sense to you when you write the report or write the paper. But if somebody is going to be reviewing, whether it's a report or a manuscript for some sort of integration, data integration, it's important for them to be able to understand what you did and why you did it, and how any variations that you can in the way you conducted the study may have altered the results from some other comparable study.
So I understand we're right at 1. And Sean, there's a lot of questions for you in the Q&A. I'm not sure that you're going to be able to get them all, but maybe we can respond to some of those and send that out. There is one that if you guys have a second to address, I think is pretty relevant. With a lot of our methods, especially going into the new approaches, we have the ability to start addressing things like the diversity of the human population, right? So sex, age, past exposures, or the genetic differences. Maybe not exactly addressing the question that was there, but what are some ways that you guys can see that we're starting to approach it, or some of the big factors that come into play? Right, because I know that Nicedum's done some work in the space, and Sean, you as well.
Well, I'll start real quickly and just say that I think there's tremendous opportunity in the NAMS space to address population variability and susceptibility of sensitive subpopulations in a way that is either just impossible or extremely resource constrained in the animal space. And in particular, when it comes to things like biomedical research, having the ability to make patient-specific models and to actually get, and create iPSCs from individual patients to understand how they will respond to personalized treatment strategies is something that is, we're just at the tip of the iceberg on that. And I think that field is going to blow up. And I think that we're going to learn a lot from applications of patient-specific models in the healthcare space that can then be carried over to the toxicology space to help us design population-based NAMs that will represent sort of the diversity and the sensitivity of susceptible subpopulations. So I'm really excited to see those two fields start to move together more in lockstep.
So Nicole, building on that, what do you think the potential role is for things like the population-based simulators? So we've got a lot of computational models now that take into consideration location, individual's location, what's nearby, as long as they're, the factors that make us human. Are those, do you see those as being helpful in doing some of the early steps, hypothesis development or exposure predictions?
Yes, and I see them as incredibly useful in more of a like a cyclical and an iterative sense. So that's one of the things that I'm most excited about with complementary is during the strategic planning phase, it became very obvious to us that where this common fund program can make the most difference is in funding integrated approaches. So taking complex in vitro methods and in chemical screens, and lab work, and integrating that with in silico predictive models, not just AI and machine learning based models, but things like these population simulators that can both assist in hypothesis generation, but then also data interpretation and hypothesis testing and evolution of those models in an iterative fashion with experimental data in partnership in an integrated way. Sean, would you like to answer the question of how many donors is enough?
That's a great one.
I know you have a lot of work in that space.
So I would, implore those that make funding decisions to realize that we need a lot more data, we need a lot more studies to answer that question. But one of the things that my group has noticed in some of the work that we've done over the last several years is that the traditional N of 3 is likely not enough to capture significant effects that are occurring in primary cell-based populations. And so, there are a number of reasons why those donor responses are going to vary. We certainly can sort of wave our hands at what those may be, but we really have to dig in to understand, is this genetic variability? Is this epigenetic variability that is derived from the donor's life? Is it something that was a technical difference in the way the cells were isolated and propagated? You can wave your hands all day about what those effects what all those things may contribute to the variability, but at the end of the day, that's a sort of put your nose down and really grind it out study that just is going to be not very flashy and be very expensive. But we need a lot more data to answer that question. But I think what has been clear in my work is that the typical ends are not likely enough on their own. And I think where the direction that's putting us to is we need to generate some of those data sets so we can understand or start having discussions with people that think in the context of uncertainty factors of it's not feasible to have studies, a regulatory study that requires 20 or 30 donors, if that's even enough, right? And so what we need to do is understand a better way that we can keep that donor number down and keep the study size practical, while also ensuring that we're collecting data to make sure that we're going to be protective of ideally the population, minimally a large part of that population, because what we want to be careful is that as we're advancing this technology in a new space, that we're not creating new inequalities and creating or using approaches and creating sort of a precedent for the use of systems that are then going to not equally protect our populations. So that's sort of the high level. There are obviously a lot of technical questions about how do we really get at that variability and how does it apply? Which endpoints do we want to focus on? Because at least in the studies that we've run, variability varies based on the endpoint. And in all likelihood, by the test article, we've only done a couple. So there are a lot of open-ended questions there.
So I realize we're getting pretty far over. I do want to thank you both for staying on and answering the questions. Contact information is there. We will have the materials sent out. I know Sean still has a lot of pending questions for him. But this is a really interesting topic, and I think there's a lot of work to be done and a lot of insight to be gained in really looking at how we validate these methods and how we, grow trust within the community to really start exploring some new things. And Nicole, I see, do you want me to tee up the link you just dropped? But
I can just mention that, there was a couple of questions about sort of follow up and how to learn more. In addition to the NICEDM website, we also have our scientific advisory meeting next week. It is open to the public. People can come in person, and if you're in the Bethesda area, or you can dial in for the webcast. And validation is a topic for next week's meeting. And Also the DNT in vitro battery, there were some questions about that. We're going to be doing a deep dive into that. So that's always a really interesting meeting. And we just released the ICVAM biennial report for 2022-2023, which is just a tremendous resource. It's, you know, a 30,000 foot view, like paragraph summary of all of the different activities ongoing in the NAMS space across the different federal agencies, both individually within agencies and cross-coordinated projects.
Thank you both very much. And definitely check out those resources. There's a whole bunch of other resources available at that website. And when the materials go out, we'll try and include some other stuff that may be helpful for audience members.
Thanks so much
So
for the
thank you both.
opportunity.
And thanks everybody for joining us and sticking up.
Thanks, everyone.